Okay guys, so today we're going to be talking about DNA replication. A few main ideas before we get started. First, the idea that DNA replication is semi-conservative. What that means is if you're starting with a parent double-stranded DNA sequence, you'll end up with two daughter strands. Each daughter strand contains one strand of DNA that came from the parent strand and one newly synthesized complementary strand of DNA. Secondly, DNA replication is anabolic, which means it's building, and that requires monomers and energy. But luckily, the triphosphate deoxyribonucleotides can serve both functions, giving us monomers and the energy to put this all together. Finally, DNA replication is complementary. What that means is that your bases always pair together. So your cytosines to your guanines and your adenines to your thymines, as we've already been taught. And also when you have RNA come in, your adenines then bind to a uracil. Do not have thymines in RNA. Next, we're going to get started talking about the mechanism here. So first, DNA replication in bacteria begins at what is called the ORIC, or more broadly, the origin. As you can see here, we have a double-stranded DNA sequence, starting with your 5' end here, 3' here, 5', 3'. Again, complementary, meaning the 5' end of one is matching up with your 3' end on the other. And how this works here is that first you have these hydrogen bonds together, holding your bases and keeping these strands together. But in order to form this replication bubble here, you need to break these bonds. The way that works is that you have a protein called a loader come in, which attaches another protein, an enzyme, called DNA helicase. What that does is it'll come in and it'll break these hydrogen bonds, moving in its own 5' to 3' direction, and that's very important because, again, it has to be complementary to the parent strands that it's bounding to. So as that DNA helicase moves forward, so as we continue down through this, your DNA helicase here will continue to move. So now it's down here and down here. DNA helicase then recruits what is called a primase, which will lay down an RNA primer, not DNA, RNA primer. So that'll attach and come following your helicase here, which would have left a RNA complementary to your bases. So here you have your bases that were and that RNA needs to happen so that we can then begin synthesis of DNA. So that RNA primer is laid down, which then allows the DNA polymerase 3 to come in and bond to that 3' OH on each side. So that will then begin laying down your DNA. So as these continue to move down the line, these hydrogen bonds will break. And if we can fast forward here, where we have, now we're going to have your RNA down here, your RNA primer is laid down here, which then DNA polymerase comes and attaches here. So you have your DNA strand down and attached. And your RNA complementary still ahead. Again, DNA polymerase does not remove this RNA primer. It simply attaches to the end of that RNA primer. However, we don't want RNA. We want a full replication of our DNA strand. So in order for that to happen, RNase down here is a enzyme that will come in and actually chew up these RNA primers. These will get removed. And so you'll be left with empty space, but as that RNA primer moves down and is eliminated by RNase, DNA polymerase 1 actually comes in and will fill in these gaps here. Again, so you have your newly strand. This would be your now replaced strands of DNA. So now you have DNA fully, no more RNA. However, what is left is a single bond here and here to be made, because DNA polymerase cannot link those final two strands of DNA. What needs to come along is ligase. So ligase is that final player there, which will seal that bond and allow it to be a fully connected strand of DNA. Next, we're going to talk about the lagging strand. So what we just talked about was the leading strand. So that's continuous. That does not stop at all as we go and continue, because it will just continue following that DNA helicase down the replication forks. So that leading strand is also called continuous. With that, lagging can also be called discontinuous. So with your lagging strand, the way those work is, again, DNA synthesis can only be made in its own 5' to 3' end. So the lagging strands can only work as fast as the DNA helicase is working as well. So with your bases here to be attached to by your lagging strand, again, DNA polymerase 3, still need RNA primase. So you need your RNA primers, but they can only move as fast as that primase. So then you end up with these small bits of DNA that are fragmented. These are called DNA Okazaki fragments, named after the scientist who designed these. So for each of these lagging strands, you have a new DNA polymerase, new RNA primase that come in. So these are a little bit more complicated, as you then have these breaks in between coming in. The important thing to remember with the lagging strand is that, again, DNA polymerase 3 can only move in its own 5' to 3' end. So with that, it can only go as fast as that bonds are breaking on either end. So you're left with these strands of DNA that each have these gaps, which then need to be brought in by ligase to be sealed in order to continue and have a full strand of DNA. And this will continue. These forks will continue until they meet at the end, because in bacterial DNA, it is circular. And the lagging strands will continue and be replaced until the replication forks actually end up meeting at the end at the termination sites. So again, your forks will meet at the ter sites or termination sites, and that will be the end of your synthesis. Continuing on, a little bit of bonus here. In bacteria, you have what are called plasmids, which are extra chromosomal pieces of DNA, meaning they're outside of the chromosome that is typically containing all the genetic material for that organism. So there's two types. The first is the low copy number, which only has about 1 or 2 copies per cell. And they'll segregate similarly to the chromosome, meaning they'll kind of split side to side when the cell splits within reproduction. And the high copy number, as you can guess, is much higher, up to 50 copies per cell, and they divide continuously. and these randomly segregate to daughter cells. So unlike the low copy, they will randomly segregate as the cell is replicated. So as the organism reproduces and will bud off and split, there's no set number of plasmids in the high copy number that will go into either cell. Next, a little bit of plasmid replication, which is a little bit different compared to what we just discussed. So there are two types, bidirectional and unidirectional. First, bidirectional is similar to chromosomal replication that we just went over. Your replication bubble with your forks going in opposite directions, leading strands and lagging strands, continuing until they reach their termination sites. However, unidirectional is a little bit different. So it's called a rolling circle model, which, as you can see by the diagram, you have a circular DNA here of your plasmid. It starts at what is a nick in the protein, which is bound by what is called a Rep-A protein, which is where the nick is bound, which is simply a break in the DNA. A 5' to 3' connection will be broken, which then will bring in helicase, which will move continuously around the plasmid and will synthesize the complementary strand continuously all the way around, while then the complementary strand will be synthesized as it is pulled off here. This is used by many bacteriophage viruses in order to replicate their DNA. And so finally, we're going to talk about the differences between bacteria and eukaryotes in DNA replication. So a few main categories here. So location, cell division, origin, enzymes, and termination. First, for the location. Prokaryotes, which we're going to have in black here. Prokaryotes occurs in the cytoplasm. And eukaryotes here, for the sake of clarity, we're going to have them in red here. And that actually occurs in the nucleus because, as we know, eukaryotic cells have a bound nucleus which contains the genetic material. For cell division, prokaryotes, that happens, your DNA replication happens during the first step of cell division. So first step. And then for your eukaryotes here, it happens during the S phase or synthesis phase of the cell cycle. For the origin, for prokaryotes and bacteria, there's only one. There's one origin, which is the ORI-C. And that's because it has a circular chromosome. For eukaryotes, it's a little bit different. There's many multiple origins. That's a result of the long, complex, and linear chromosome, so that allows for it to be read and synthesized at multiple times. Additionally, with both of these, some similarities also is that both, however, are bidirectional. Excuse me. Bidirectional meaning that they go, DNA is synthesized in both directions at the same time. As we saw above, you had a complementary strand to the 5', 3' on top, and a complementary to the bottom, both being synthesized at the same time. And both of these, both eukaryotes and bacteria, have the Okazaki fragments. No type of cell has kind of figured out how to get rid of these in terms of replication. Plasmids are a little bit different because it's very simple, just that rolling circle. However, bacteria and eukaryotes both have those lagging strands that can only be synthesized as fast as the helicase moves. Also, for enzymes, prokaryotes here, again, we'll use DNA polymerase 1, which is what replaces the RNA primer. And we'll have DNA polymerase 3, which is what lays down the leading strand. And eukaryotes are a little bit different and a little bit more complicated. So eukaryotes use DNA polymerase alpha, delta, and epsilon. In terms of termination, prokaryotes, again, these are for your eukaryotes. Termination for bacteria have a single terminus. As we discussed, they both continue, the leading strands will continue and eventually meet together at the ter site. And for eukaryotes have several termination points. Because, as we said above, it has many origins, so they need to have many termination points. So overall, that is the way that DNA replication works in bacteria and some differences between bacteria and eukaryotes. Thanks for staying with us and look forward for more videos.